Hi everyone, welcome to Bio 1500 Ignite Talk. I'm Jing.
My name is Bozo.
And I'm Chunlu.
So today we'll present on the antibiotic resistance module review. And first, I will start with the important concept evolution. So evolution is changing the allele frequency within a population over time. And it is promoted by two methods, so from natural selection or artificial selection. And natural selection is happen in a natural condition, which certain traits are favored than other and pass to the offspring, which helps those species survive better. And the example here is the giraffe. So giraffe has longer neck that can reach the leaves better, will pass this trait to their offspring. This is an example of natural selection. And for artificial selection, which is basically humans artificially select a traits that will be preferred and to certain species. So here the example is the corn. And in the experiment, we did the antibiotic resistance on bacteria, and the mutation creates the variation of different traits. So like the charts on the right shows like before selection, the mutations cause a bunch of different traits of bacteria. So here, for example, like if the bacteria have different pigments, then after the mutation happens, those bacteria with like red pigments will be favored and pass to the offspring, and the other colors will be selected against, which will not be passed to the offspring. And eventually, in the final population, the bacteria will basically just have the red pigment, which also means bacteria with red pigment will have higher resistance levels than those with other colors.
So now I guess it's important to know a little bit about how bacteria grow. So bacteria grow basically by binary fission, and that's why their growth is expected to be exponential if resources is not limited. So when you are actually growing a bacteria in a limited resources environment, there are 4 stages of their growth. The first stage is the lag phase, which when you at the beginning, when you first planted the bacteria into a plate, they need to accommodate to the environment a little bit before they can grow and divide. The second stage is the log phase, in which the bacteria grow exponentially as expected. And the third stage is the stationary phase, because as bacteria grow, the resources is less and less available for each bacterial cell, so they die. And when the death rate becomes close to the division rate of the bacterial cell, the number of cells in the culture stays stationary for a while. That's the stationary phase. And the last stage was the death or decline stage, because ultimately you're getting less and less resources, and the bacteria cannot grow in your environment, so they die and die out. And there's just a formula for estimating the number of cells in the culture, n equals n0 times 2 to the power of small n. The n0 here is the initial number of cells in the culture, and the small n here is the number of generations you have passed, and eventually you get the number of cells in your final population, which is this n. And So there are two different models or hypotheses about how mutation rises. And the first model is induced mutation, which basically says that mutation doesn't arise until you apply some selectional pressure onto the population. In our case, is the antibiotics, because antibiotics are known to kill the bacteria. And in this model, mutation rise almost, since mutation is random, they almost arise in equal number of cells in each culture after applying the antibiotics. But in the second model, which is the spontaneous mutations, it's hypothesized that the mutations basically arise randomly without any need of any specific environmental conditions. So if one cell's mutated early in their lineage, sorry, can I go to that stage? If one cell is mutated early in their lineage, then all of its descendants will have the mutated phenotype. And in that culture, after applying the antibiotics, you see a lot of cells with resistance. And in some cases, some cells don't mutate at all. Then in that culture, after applying antibiotics, you see no resistance. So there, in this model, it's expected to have you're expected to see a lot of variations in the number of cells that survive the antibiotics, and that's actually what we see in the experiments.
So here are some general background knowledge about antibiotics. So it's originally referred to the substances produced by one microorganism, which is inhibited the growth of the other one. And it acts against different groups of bacteria to inhibit the growth of the bacteria. So there's generally two types of bacteria. One is gram-positive and the other one is gram-negative. There's like difference between their cell walls. So the gram-positive bacteria has generally a thicker layer of peptidoglycan. So this one affects the sensitivity and resistance to some types of antibiotics. And the two different antibiotics we use during lab are penicillin and streptomycin. So penicillin is a beta-lactam antibiotic. So it functions by targeting the bacterial cell wall synthesis. And the penicillin is more effective against gram-positive bacteria. And the streptomycin is an aminoglycoside antibiotic, and this one functioning by inhibits the translation, which prevents protein formation. And streptomycin is effective for all types of bacteria. So I'm going to wrap up with a refreshment about what we did during labs. So we used either the cell count, colony count method or the disk diffusion method to do our project, which compare like different traits and variables about antibiotic resistance of bacteria. So the colony count method count the numbers of cells forming the colonies on the plates. And the disk diffusion method calculate the average zone of inhibition. And here are the two figures that shows these two methods. So thanks everyone. I hope you enjoy this module.